Breaking news. Hold on to your capes. In a thrilling twist straight out of a comic book, scientists have unleashed CRISPR, the superhero of gene editing. Armed with precision lasers guided by a GPS-like system, CRISPR is here to save the day by rewriting our DNA with lightning speed. Our hero CRISPR uses a sidekick guide RNA to pinpoint the exact location of genetic flaws. Imagine gRNA as a mini-map that leads CRISPR's laser-like Cas9 protein directly to the target. The DNA is cut and our hero's trusty repair crew swoops in, fixing flaws, erasing harmful genes, and even adding super-powered new traits. This epic battle means CRISPR can cure genetic diseases, bolster crop defenses, and turbocharge scientific research. Our hero's ability to make precise genetic edits is turning the tide against genetic disorders and ensuring a future brimming with possibilities. So join us as we cheer on CRISPR, our genome guardian, in the fight against forces of flawed genetics.
This is how CRISPR works. A guide RNA leads the Cas9 protein to a specific part of the genome where it cuts the DNA or RNA at a precise location. The virus's defective genes are either repaired or deleted, rendering the virus unable to replicate or infect host cells. The best part, it's incredibly specific. Unlike broad-spectrum drugs, the superhero technology CRISPR can target just a mutated region of the drugs, leaving the rest of the genome untouched. In lab tests, we've seen how CRISPR can stop viruses like this one in their tracks. We use a nanoparticle delivery system to carry CRISPR into the infected cells. Once inside, the system edits the viral RNA, preventing further replication. This method is fast, precise, and adaptable, capable of evolving the virus itself. But this is not without its challenges. The technology is powerful, but it is not perfect. We need to ensure that CRISPR doesn't cause unintended edits to the host genome. We need rigorous testing to make sure we're targeting only the virus and not the patient's own cells. CRISPR is a superhero, an incredibly precise one. It's not just for curing diseases, it's for preventing them, rewriting the very genetic makeup of life itself. The potential is enormous. With great power comes great responsibility.
Another thing CRISPR can be used for is plants. Because we can precisely add or remove genes, we can selectively change aspects of plants, such as the texture or flavor in the salad. Maybe it's crunchy, maybe it's bitter, maybe you want one of those and not the other. Traditional plant crossbreeding methods involve basically mixing two plants together. And now with CRISPR, we can modify plants in the most specific way possible. Plants have immune systems too, and we can use CRISPR on them to make them more resistant to diseases and fungus. You can happily enjoy your salad made from the happiest, healthiest, tastiest plants.
What then can be said about CRISPR for the future of humans? We can use that to modify the genomes of embryos to ensure that they aren't born with some diseases which include sickle cell anemia and even HIV. So we do believe that there's a bright future for CRISPR for both animals and plants.
Looking ahead, CRISPR holds great promise for the future. Beyond agriculture, researchers are trying to find ways plants could withstand immense weather conditions, genetically engineering plants that could absorb more carbon dioxide from the air, so that they could adapt more readily to steady shifting climates. Additionally, this technology could be used to fortify the nutritional benefits of crops, which helps with overall global malnutrition. As CRISPR continues to evolve, we must maintain a balanced perspective, fostering innovation while establishing general policies and ethical guidelines to navigate potential changes. Back to you.
Although CRISPR introduces incredible possibilities for our future, there are several ethical concerns from scientists. Editing human embryos or germline cells could be genetically passed on to future generations. You can edit the genes of all these unborn babies and design it how you please, but the baby and their future generations would not have been able to consent to this gene editing. It raises ethical concerns about autonomy and individual rights. The gene editing could not only prevent genetic diseases, but enhance features such as intelligence, physical appearance, or even behavior. You could create your very own super baby, which could limit individuality from the human race and would cause social inequities and discrimination based on genetic privilege. The discrimination would come from societal standards and place barriers between those who have been genetically enhanced and those who have not. These CRISPR treatments would most likely only be available for the wealthy, therefore causing a further divide in economic classes across the nation and across the world.
As we near the end of the plot for the CRISPR hero and his sidekick, the gRNA map, he proved victorious in his fight against the genetic villains. CRISPR was able to use his Cas9 laser beamer to finally put an end to the genetic threats. Because of CRISPR and all of his components, humankind is now able to reach new heights. Let's all thank CRISPR and of course gRNA, our brave protectors, to allow us humans to achieve smarter and healthier futures.